Awesome. Stupendous. Did anyone have any questions? Before we start with today's lecture. But that's not Monday. That's not Monday. That's today. Put everything away. Let's take our test.
It's next Monday.
Are you sure? You don't take it today?
No.
I think today was us information on it. I would also have to make it up in my head. It would be multiple choice. It'd all be short answer. Let me get out of there. Okay, who's ready to talk about DNA replication in the most amount of detail possible? Anybody? Wait until the light up. Because this is genetics, not one biology. So we're not going to focus so much on mechanism of replication. Our main focus on replication is because of this idea of fidelity. And some people may be familiar with the fidelity in relationships. And what does infidelity mean in a relationship? Cheating, seeing someone that's not your partner. And that's an important idea about replication fidelity. Everything has a partner. Base pairs. A goes with what? You good guys? Who remembers the base pairings? A with what? B, and then G would go with what? C. So if you remember our base pairing rules, that really help out because it's important. What won't be that really important is directionality. At least for us right now, directionality is not that important to talk about. Although we can't talk about DNA without talking about the strands of DNA are directional molecules. It does impact us a little bit, but not as much as you might think. And again, when we draw a double-stranded DNA molecule, one of the most important concepts of the DNA molecule is the idea of anti-parallel. So the two individual strands of the DNA molecule go in opposite directions. And we typically denote this by drawing arrows. And while the phosphate group and the hydroxyl group are not really important, the 5' end and the 3' end are important to think about because we need to think about directionality sometimes. And which direction something goes tells us if we are adding or removing nucleotides, which will be an important part of what we're going to deal with is, are we adding things or are we removing things? And what direction do we need to go to do one of those two things. Well, anti-parallel is important. The other really important idea is base pairing, which again we just talked about briefly. But an A in one strand base pairs to a T in the other, a T in one strand base pairs to an A in the other, and similarly long G's and C's base pair together. So this is our basic picture of a DNA molecule. Anti-parallel strands, complementary and nucleotides. So base pairing and complementary, A's to T's, G's to C's. Again, there's a lot of people that worked on the structure of DNA. Watson and Crick get the most credit for determining the structure of DNA. Probably can't forget Franklin and Wilkins contributed a significant amount of information to figuring out the double-stranded anti-parallel nature of the DNA molecule. And then Erwin Kargath also giving us ideas of base pairing rules before we knew about what base pairing actually was. So again, a lot of important scientists gave us this picture, even though for exam 2 we don't care about those people. They were only in exam 1 type of thing. For this test, this idea is not as important, but it will be on the test. So, but for exam 3, this idea of purines and pyrimidines are going to be much more important. Now you may remember there was a question on exam 1 about purines and pyrimidines. There'll be a question on exam 2 about purines and pyrimidines. There'll be a question on exam 3 about purines and pyrimidines. And most likely on the final. It's a pretty important concept. Purines versus pyrimidines. So as we look at our nucleotides, the purines are A and G, and our pyrimidines are T and C. And so in every base pair that happens, the purine-pyrimidine base pair, this large versus small joining together is an important concept we want to keep going forward A's and G's, the big nucleotides, C's and T's, the small nucleotides, and that makes it fit together in the appropriate way. If you care about knowing what the base pairing is, and in other classes you may be asked to draw the nucleotides and how they base pair together, that's not something that we care about. But they do base pair together in appropriate and predictable ways, and because of the way in which the nucleotides are arranged and the groups that are on the nucleotides, A is only going to base pair naturally with T, and G will only naturally base pair with C. This is a pretty important part of why we are talking about replication. Nucleotides will naturally base pair with other nucleotides, which means they're not supposed to base pair with other nucleotides. And that gives us an ability to spot when things are wrong, when things are not doing what they're supposed to be doing. Now, discovering, or at least elucidating the three-dimensional structure of DNA is a pretty cool thing. One of the more important ideas that came out of Watson and Crick's paper was this idea they threw in kind of the end of the paper, almost a throwaway statement. And they talk about DNA being complementary, and they hypothesized that because DNA is complementary, a very easy way to copy DNA is to pull apart and slap on new DNA. Because base pairs are going to base pair together in the appropriate way. Just sort of let it happen. Because naturally things sort of go together. When it comes to showing how replication actually works, we have two scientists, Messelson and Stahl. And we're going to talk about their experiment, but I'm not going to ask you to memorize all the nitty-gritty details of this particular process. There are some important things I want you to think about. DNA is a big thing. Messelson and Stahl use bacteria as their model organism. Again, most of the things we know about replication, transcription, and translation came from studying bacteria first, and then applying what we knew about bacteria to see if it's also true in eukaryotic organisms. And it turns out that molecular biology is basically the same. How you make DNA, how you make RNA, and how you make proteins are generally a universal thing that everybody does similarly. But when you're studying really big DNA molecules, you need to have a way to sort of study new pieces of DNA and old pieces of DNA. And what Messelson and Stahl decided to use were nitrogen isotopes. And so they started with DNA being full of one nitrogen isotope, and then grew the cells until all their DNA had that one nitrogen isotope. Then they gave those cells a different nitrogen isotope and see what they did with it. So the idea of this experiment is if all the DNA is one type of isotope, it's going to be either heavy or light, because 14 is lighter, 15 is heavier. As you get the new nitrogen, we're going to see a change in the DNA size as new DNA is being made. Now again, you don't need to understand all the different details of how this works, but Messelson and Stahl used size differences to study the replication process. And instead of using DNA gel electrophoresis, they used something else, which is called cesium chloride gradient ultracentrifugation. It's a pretty long, complicated idea, but centrifuges separate things by size. So if something really fast, things separate. Big things, heavy things travel farther, and smaller, lighter things travel less far. Basically, a centrifuge is the reverse of electrophoresis. Remember, electrophoresis, big DNA molecules were slow, didn't travel very far. Smaller DNA molecules travel much further, much faster. But here, the bigger you are, the more you are moved through the centrifugation process. So you can easily separate things by size. New DNA and old DNA can be separated by size. Again, what we're going to see in this experiment is the cells start with heavy DNA, and over time we're going to see what happens to the DNA as a lighter version of nitrogen is incorporated into the molecules. Before we look at the data, Messelson and Stahl had three ideas of what they thought were possible for how do we copy DNA. One was called conservative, the other was called semi-conservative, and the third one was called dispersive. We don't really need to know all the different possibilities. But based on the data they saw, it what's the word? Demonstrating, I guess it's not the word either, but here we see a pattern that matched what they expected for a semi-conserved replication process. I mean, don't need to look at all the different parts, but if we just look at a few parts here, one of the things I want you to think about, all of our DNA starts in one location, and as we go through one full round of cell division, meaning we've copied our DNA, you see that the DNA is now at a higher or lighter weight. So something is going on. And as you proceed through another round of replication DNA, we go from having one band now to having two bands. And only one of Bessel and Stahl's models predicted this as an outcome. As you keep going further from this one to this one, you still see the two bands, but the bottom one gets lighter, the top one gets darker. We go one more round, the bottom one gets lighter again, the top one gets darker and again, still darker. So all of this feeds into one simple idea that Bessel and Stahl called the semi-conservative replication process. And that's what we call replication. It's a semi-conservative process. Semi-conservative means that when we duplicate our DNA, we take one DNA molecule and get two DNA molecules. And those two DNA molecules at the end are half old and half new, or we call half parent and half daughter. That's why it's semi-conservative. We save half of the original molecule and then add in half of the new molecule. So what Bessel and Stahl were seeing in their experiment was a change from heavy to light over time because of the way replication works. So we go from two red to half red, half blue, and then separate the blue from the red and get another new blue one, so half blue, half red, and then all blue. And eventually you get more and more of these all blue molecules, which are we consider the light molecules. Now what is here is more than you need to care about. This is the easiest way to think about semi-conservative replication. And this is, in a nutshell, what happens during the replication process. One DNA molecule is pulled apart and used to make two new DNA molecules. So the old piece is separated, and then we make two new pieces that are half old, half new. And there's a little bit more that goes into this process, but this is what we do. We pull apart the DNA and we add in new DNA to it by simply following base pairing rules. Now for us, there are a few places where we're going to think about, mostly because these are places where mistakes need to be either dealt with or they're going to be introduced during these different parts. So again, one DNA molecule is pulled apart, and then we get two new, or two double-stranded DNA molecules at the end. And this is what semi-conservative process means. When we make our DNA, they're half old, half new, and then new double-stranded DNA molecules. This doesn't tell you how it actually works, just as what the end product is relative to the starting product. Semi-conservative is beginning to end and not worrying about what happens in the middle. And when we worry about what happens in the middle, we have different ideas and different names to describe the process that goes on between beginning and end. And it's a place where some people get confused. Semi-conservative, again, just refers to the starting molecule and the two ending molecules. But as we look at the actual process and how it works, there's another name that we use instead. So one DNA molecule separated, pulled apart. That's what we call the bubble formation step. And to make the new DNA, we need basically all of these other steps involved. It looks fairly complicated, but we're going to try to simplify it a little bit. And one of the really worst sort of complicating parts of replication is that's replication, making DNA. Turns out making DNA is not so easy. And it'll complicate our issues as we go forward. When we look at the actual mechanism of replication process, we call it a theta replication process. Because a structure that looks like the Greek symbol theta is formed during this process. And because we studied bacteria first, the theta symbol was very recognizable because we start with a circular piece of DNA. So theta replication is semi-conservative replication. All replication is semi-conservative replication, but not all replication is theta replication. It's sort of an important distinction. In semi-conservative just means beginning versus end, what do our end molecules look like versus what do our starting molecules look like. Theta tells you basically how does it work in the middle. And keep that in mind. And when it comes to using theta replication, every single chromosome uses theta replication as the way in which we make new DNA. Both eukaryotic linear chromosomes and prokaryotic circular chromosomes all use theta replication as the mechanism. There's a picture in here. No, let's keep it in here. Let's go focus. No, take it out. Just draw you a little picture. Copying a circle is not really that much different than copying a straight line. So if you think about doing the replication process with a circle, we form a bubble that sort of starts out this process. Same thing is true when we look at a eukaryotic chromosome. But one of the things we will notice and hear about very quickly is the number of starting locations is going to be different. And so in each of these two cases, we form something that kind of looks like a theta. I know it doesn't look like theta, but that's the idea. So when we copy our chromosomes, we are forming something that looks kind of like this as a starting point. Whether it's a circle or a line, we're forming these sort of bubbles that make the replication process work. These bubbles will move from their starting location in a bidirectional direction. Bidirectional direction. Direction was right there, but they move sort of laterally away from their starting point. And one of the cool things about replication being bidirectional is it makes it happen faster. And so you can imagine you're traveling around a circle. If somebody's going in the opposite direction, you're gonna meet each other sort of at the end or at the other side. Each thing goes half the distance. If you only have one direction, you would take twice as long to get around to copy the whole thing. Same thing is true for a eukaryotic chromosome. You have these things moving in opposite directions, trying to copy all the DNA as fast as possible. And that's really all that's going to matter when it comes to counting or looking at how many starting places there are. The more DNA you have, the more starting locations you have. Because you have more DNA to copy in a shorter amount of time. Now, chromosomes are not the only thing that will use the theta replication mechanism. There are a lot of plasmids that will also use the same basic mechanism. One of the things that's weird is some plasmids will use a different mode of replication process. It's still semi-conservative. Take a double-stranded DNA molecule, make two new pieces of DNA, half old, half new. But the how is different. And so some plasmids will use what's called a rolling circle replication method. And this is particular for plasmids that will only be moved through the process of conjugation, which is a form of bacterial sex. There's a structure involved called a pilus. It connects two cells, and this DNA is transmitted through this protein structure from one cell to the next cell nearby. How it works is not important. It is just showing that replication can happen in different ways. But when it comes to chromosome replication, there's only one name of the game, and that is what's called theta replication. But sometimes chromosomes or plasmids, we use this alternative version of what we call rolling circle replication. It's pretty weird, but we don't care how it works. In molecular biology, you probably hear a little more about how it actually works. It's not important for us. The mechanism of these things. Now, what is on this slide is something that I showed you on the board behind here. Because of the size of the procreative chromosome, you typically manage to copy that whole thing using one starting location, which is what we call an origin. Sometimes it gets a more formal name, origin of replication. A lot of times it's called the origin. Where does replication start? Where does that bubble start first? In a eukaryotic chromosome that is significantly bigger and longer than a prokaryotic chromosome, we have multiple origins. Now this 8,500 is total over all of the chromosomes. Typically it's like a human number rather than another number. But we have 23 pairs of chromosomes, that's 46 total pieces of DNA that have to be copied every time our cells divide. And to copy all those, you at least need 46 origins, one for each chromosome. But some of them are really big, they need multiple ones to actually work. And origins just give us faster replication. That is the only thing that an extra origin is going to give us, is faster replication of DNA. Typically, replication of DNA is tied to how fast your cells divide. It's really not going to be any, I should say, you can probably forget that whole idea because it's not genetically related to us. But replication speed is tied to how fast do cells divide. If you can slow down replication, you can also slow down cell division because those two things are sort of tied together. And that's something that will be important going forward is this link. How fast do you copy your DNA? How efficient do you copy your DNA? It is directly tied to does cell division function normally. If there's a split between the seams, if replication is going wonky, then that means cell division is also going to go wonky as well. So we do need to sort of tie those two things together. Cell division and DNA replication are both needed. Just to go back for meiosis, what do we call these duplicated copies of DNA?
Sister chromatids.
They call them sister chromatids, and they're paired because they're stuck together. So as we copy our DNA, remember that new copy is stuck to that original copy, or vice versa. Because it's not the original, it's half old, half new, plus another half old, half new piece. And they stay together until metaphase, or actually anaphase, right? That's when we have to separate things. But those DNA molecules stick together. That's sort of one of the places we see a difference between prokaryotic organisms and eukaryotic organisms. We don't have sister chromatid pairs in bacteria. When the DNA is pulled apart, when we see bubble formation, we do see the stress and tension being introduced into the DNA molecule. For both a circle and for a linear chromosome, the ends are not free to rotate. They're sort of stuck together in a place where they can't sort of rotate around each other. And that creates tension. I know maybe you're too young to know what a phone cord looks like. Raise your hand if you've seen a phone cord. Yeah, maybe not that young. What happens when you sort of put the phone back onto the handset, back on the receiver part? The little cord sort of coils together, right? You can unwind it and you can get rid of those coils. So that's the idea of tension. We call this sort of supercoiling in a sense. But we call it tension. Hopefully you can kind of see that the peaks and valleys get closer together as you pull apart this thing. And if you have too much tension in the DNA molecule, you can't do anything with it. We need to relieve tension. Now we can't take a good massage, but we can do something else, which is cut the DNA and unwind it. And typically you think about the bubble as an unwinding of the DNA, but we still need to unwind the DNA after unwinding the DNA. We need to relieve tension, because everybody's under stress after a day's work. The enzyme responsible for relieving tension is called a topoisomerase. What's the definition of an isomer? Remember some chemistry or biology 140?
Same molecular formula, but different arrangement.
Same molecular formula, different arrangement. If you change the shape of DNA, the DNA is still there. You're changing the way it looks. And what is a word that starts with topo that relates to shape? And you know, a word, topography, topology, similar ideas, shape, elevation. So the name literally means changing the shape of DNA. But for us, to relieve the tension, we are going to cut the DNA. This is where an issue can come out. Cutting DNA can cause problems. So anytime you operate on somebody, there's potential for something to go wrong. You open somebody up or cut the skin somewhere, there's at least some possibility of something going wrong. So when we cut our DNA, there's a chance that it might not go back together correctly. Things might get lost, things might get gained. Same thing is true for crossing over. Remember, recombination is cutting the DNA. But we're cutting it twice and swapping the DNA for the crossing over process. Here we're cutting DNA not to cross over, to recombine. We're doing it so we can unwind and relieve tension. Turns out there are two types of topoisomerases, a 1 and a 2. The 2 cuts both strands of the DNA molecule. The 1 only cuts one strand of the DNA molecule. And we're going to care about the number 2 because the enzyme needed for replication is called DNA gyrase. It's kind of a fun thing. DNA gyrase is going to bind the DNA and then cut it and kind of unwind it like you would turn a crank. But as it's sort of doing this thing, it has the potential to not put it back together correctly or something could go wrong. If DNA gyrase is malfunctioning, cutting the DNA could cause issues. So we need this thing to be functioning correctly. If it doesn't function correctly, then DNA replication can't work. Without this happening, replication is stopped before it really even started. So cutting the DNA, but cutting both strands, is an important thing about DNA gyrase. The other important idea is this is an epigenetic process, meaning it does not care what the actual DNA sequence is. It has to be able to cut every piece of DNA, regardless of what it says. Because we need to copy the entire chromosome from end to end. So we need to cut this place a lot of times. And it happens fairly often. Cut, you unwind, and then you open it up to cut and unwind again. You just keep doing this the whole time. You're copying your DNA. We're cutting DNA quite a bit and putting it back together quite a bit. This is a unique class of enzymes that we talk about a lot this semester. Things that cut DNA. And one of the things you'll be asked to remember, how many times is it cut? Is it sequence independent or dependent? And one other thing that's related to how many times it cuts as well. We'll get to that as we go forward. We got this bubble. The bubble is where we start to pull apart our DNA so we can base pair new DNA to that old DNA. It turns out the enzyme that makes DNA is a shitty enzyme. I wish I could say it's worse than a shitty enzyme, but it's a shitty enzyme. It can't start making DNA on its own. It requires a starting thing. That starting thing is called a primer. Now the enzyme that makes primers works really well because the primer is RNA. So the enzymes that use DNA to make RNA, like transcription, very easily can start making RNA anytime, anywhere they want to, basically. It's one of the weird particular things. You can make RNA without needing a starting molecule, but when it comes to making DNA, a starting molecule is required. It's what we call a primer. And the fact that the primer is made out of RNA is why we have a later step where the primer is removed. We want to get rid of all the RNA, so our molecule is just DNA. So there's a link between this step and the second to last step. Make an RNA primer, remove the RNA primer later on. One of the underlying themes of this entire section here is that eukaryotic organisms suck. They are complicated. Bacteria, are much simpler to deal with. One example of that complexity versus simplicity is that in bacteria, like prokaryotic organisms, we have RNA primers that are pretty short, and they're only RNA. But when it comes to eukaryotic organisms, the primers are much longer, about 36 nucleotides versus 6. And the eukaryotic primer is both DNA and RNA. So the question is, I put to you, why? If it's good enough for bacteria, why is it not good enough for eukaryotic organisms? Even having us, why do we need extra long primers that are both DNA and RNA? Like we use a small RNA primer to make a short DNA piece, but then we stop and don't continue until we come in and grab onto that short DNA piece to make it a much longer DNA piece. This thing is stupid, anybody else? Like, why start making DNA only to stop? Only to start making more DNA later on. I can't think of a reason why it works like this. This just seems dumb. But this is the way it works. Unfortunately, for you, prokaryotic organisms are simple and eukaryotic organisms are complex. We see differences in origin, we see differences in primers, we see differences all along the line. It's just the steps are the same, it's just the components are a little bit different. When it comes to doing the semi-conservative replication process, DNA polymerase is the main enzyme, because it's the thing that's going to make our leading and lagging strands. We don't necessarily care about being able to differentiate leading and lagging much in this class, but there is a main polymerase that makes the majority of the DNA during replication. And there are other DNA polymerases that do other things for the cell. So there's a main DNA polymerase used for replication. We call it DNA polymerase 3 in bacteria, or DNA polymerase delta for eukaryotic organisms. Why? Why can't eukaryotic organisms use the same language as prokaryotic organisms? Bacteria are here first. Why can't we use their naming system? Why? Ego. I found something, I must name it. Right? This Christopher Columbus, everybody, I claim this in the name of Columbus. Or whatever, Lee Harrison, thumbs are here. But one thing we do see in all organisms, both prokaryotic and eukaryotic, is the presence of multiple DNA polymerases. And one of them is, or two, are the polymerases that are used for the majority replication process. The others are used for other things. Now, some of that other stuff is replication related. The other is repair mechanisms, where we're going to spend a lot more time on for exam 3. But we'll see some repair mechanisms here for replication specifically. We got a lot of polymerases. They all make DNA in basically the same way. It's just some of them have different actual purposes inside the cell. For both prokaryotic organisms and eukaryotic organisms, the DNA is broken into different parts because of the directionality of the replication machinery. We get one really long piece called the leading strand, and a bunch of really small pieces called the lagging strands, which are called Okazaki fragments because of the scientists that first viewed them. There is a weird type of thing we see. The size of these Okazaki fragments does differ. And basically, eukaryotic organisms make really short ones, whereas prokaryotic organisms make fairly long ones. Is it more complex to make something longer or make something shorter? I don't know. What do you think? What's more complex? Make something longer or make something shorter? Well, apparently not, because bacteria are the simpler type of organism. Now maybe there's other things going on that we don't necessarily see behind the scenes. But there's a weird thing going on. The replication is not exactly the same. It's still semi-conservative, It's still theta replication, but the nuts and bolts underneath the hood are a little bit different in how we do things. So we use one main polymerase to synthesize these leading strands and lagging strands. Doesn't matter how long they are or how many there are, we're gonna copy our DNA. And before we're done, we do need to remove all those RNA primers and replace them. But as we're making the leading strand and the lagging strand, we are going to check our work. That's the first place where we're going to see fidelity become important to deal with. The DNA polymerase, the main DNA polymerase, has a proofreading function. It's a way to detect if an error has been made. Proofreading is basically spell check for you. It's kind of automatic, it's immediate, happens right away, and you basically don't worry about it, but you just keep going, because it works fairly efficiently. If we look at the polymerase without the proofreading activity, and that's something you can do, you can mutate the proofreading activity without affecting the DNA copying activity, there's an error rate for the enzyme itself. So replication by itself makes a mistake 1 for every 10 to the 5th nucleotides that are added. Like 1 in 100,000. That's not bad, right? 1 for every 100,000. A bacterial chromosome is about 4 million base pairs. Eukaryotic chromosome, maybe 100 million base pairs. 1 in 100,000 look pretty good when you have 1 million things to do, or 100 million things to do. No, not really. You're gonna make a lot of mistakes. If you have to copy 1 million things and you make a mistake every 100,000, that's a fairly large number of mistakes. Maybe it's 100, but still, maybe it's not that many, but still, hey, 1 is bad. That's what we're trying to get down as sort of 1. So the polymerase by itself has a certain error rate. If we add in the proofreading activity, we can increase our fidelity and decrease the error rate. So we call this the proofreading activity of the polymerase. It's called 3' to 5' exonuclease activity. When the enzyme is making the DNA, it's going in the 5' to 3' direction. And so the fact that we go in the opposite direction is important. The enzyme stops and backs up. That's how proofreading activity works. You make or try to base pair, add a nucleotide, and if you're right, keep going. If you're wrong, you back up and try again. What does nuclease mean? Anybody have any ideas what nuclease might refer to?
Breaks apart nucleotides.
It's an enzyme that breaks DNA. Exo tells you, is it on the end or in the middle? So exonuclease removes nucleotides from the end, going in the opposite direction. If the polymerase breaks in the right way, it can add a nucleotide and remove it in an endless cycle and never go anywhere. Because adding it as soon as it's adding, removing, and going sort of nowhere. But luckily, it usually doesn't break, so it makes a mistake and fixes it and goes on really quickly. But we have to cut the DNA. The DNA polymerase is a nuclease. It's going to cut DNA. But rather than cutting both strands of the DNA molecule, we only cut one strand. So here we have already 2 DNA cutting enzymes. DNA gyrase, the topoisomerase, cuts both strands in a single location. But the proofreading activity of DNA polymerase will cut one strand in any number of locations, depending on how many nucleotides there are coming along. But it's sort of a one at a time deal. If you make a mistake, you back up and you try again. What's the probability of getting the right nucleotide in here? You guys should be good at probabilities already. It's 1 in 4. There's 4 nucleotides to choose from. A, T, G, C. 1 is one of those 4 that's right, so you got a 1 in 4 chance of getting the right one each time. But DNA polymerase is so good that it does it 99,999 out of 100,000. So it's not random, which is the important idea of DNA polymerase replication. It's not a random process. It's not randomly picking something. It's pretty good at picking things. If he gets it wrong, removes it, what's the probability of picking the next nucleotide correctly? It's 1 in 4 again. Every time you add a nucleotide, there's a 1 in 4 chance you're going to get the right one, and a 3 in 4 chance you're going to get the wrong one. Which I think is interesting to think about. The probability of getting the wrong one is the more likely event to happen. But the right one is picked, 99,999 out of 100,000. That's a pretty good probability. So there's something going on. I think it's pretty neat. Now, because of proofreading activity, we go from 10 to the 5th, or 10 to the minus 5, to 10 to the minus 7. So we've increased our fidelity by adding in this proofreading activity. And I like to think about this in parts. The polymerase has a sort of a fidelity rating by itself. When we look at the replication itself, that's 10 to the 5th. If we think about DNA polymerase plus proofreading activity, we can get down to 10 to the 7th. And so this gives us basically a 10 to the minus 2 contribution. Let's go from 5 to 7, 10 minus 2. Now there's one more thing that we're going to add in, but we're not quite there yet. But there is another thing we have to worry about, which is called mismatch repair. And that's going to get us down to 10 to the minus 10, which is another 10 to the 3rd contribution. And make a mistake every, what is that, 10 billion? One mistake in 10 billion chances? You take those odds. Imagine how good your grades would be if you only made a mistake once every 10 million things you did. You'll never make a mistake, basically, for your lifetime probably. But proofreading activity is this exonuclease activity that is immediate. And we're done. But it's going to affect the synthesis of the leading strand, and it's going to affect the synthesis of the lagging strand. Now, unfortunately, proofreading activity does not make replication foolproof. Mistakes still get made, which is why we have this next thing going on to catch any mistakes that proofreading activity missed. Before we get to that, we have to go on to the next step, which is the removal of the primer. Remember, the primers are RNAs. We need to get them out of our DNA molecule to replace them with DNA. This is where those other DNA polymerases come into play. We're going to remove the primers and fill in the missing area with DNA. And at the very end of the process, there's an enzyme called DNA ligase, which will form phosphoester bonds to connect everything all together as one continuous piece from end to end. Now, ligase is important. We're going to see ligase quite a bit, but we kind of forget about it as we go along. But when you're looking at this one, it says another DNA polymerase, right? Not the main DNA polymerase, not the thing that makes the leading strands and the lagging strands, not the thing that has proofreading activity. Because what we've learned is that usually these things don't have proofreading activity. But they only make a little bit of DNA, so the chance of making a mistake is pretty small. If they make a mistake, we still have something later on that can maybe catch any of those things. Again, proofreading activity is an immediate sort of spell check. Who's been fucked from spell check before? Who sent a message to somebody like, oh crap, what did I say? You get a little question mark back. So what would fix that would fix those types of mistakes? In your life, how would you fix that type of mistake before it got sent? Come on, you guys know it, it's an easy thing.
Autocorrect.
Well, proofreading activity is autocorrect. Sometimes autocorrect picks the wrong word, right?
We read it over again.
And reread it. That's what mismatch is. Rereading the DNA to see if there's any mistakes. But luckily, the base pair is a certain size. The A to T is a certain size. The G to C is the same size. So if you have A with C, it's not going to form the right kind of size that A and T will, or G and T won't form the same size, or A and G looks weird, T and C looks weird. If you have the wrong base pair going on, there's a noticeable sort of difference in how the DNA looks. Again, with proofreading activity and mismatch repair, we get a pretty good fidelity rate. One mistake every 10 to the 10th nucleotides that we add. Again, this is more of an editing function, a rereading process, to check to see did we miss anything. And as you can see, mismatch repair relies on DNA methylation states. And we're not going to finish all of this before class ends, but we'll see how far we get. And what I want you to think about is, we'll go back to our basic picture of replication as a starting point. And as we go through replication, we have a DNA molecule, which we draw like this. 5' and 3' don't really matter, but as we go through the process, we separate the old strands from each other, and then we make our new piece of DNA. So if we have an A here, hopefully there's a T here. There's an A here, what if there's a C here instead? The problem is, how do you know which is the correct nucleotide when there's a problem? Is the A right or is the C right? If you could guess from the other side, but maybe you can't. The polymerase can't see the other side of DNA at all. The enzymes that are dealing with mismatch repair can't see the other thing at all, they just see this. And how do you determine which one of these is right? And that's where the methylation of DNA is going to be important. Methylation of DNA allows us to tag the DNA as old versus new. So as we start, our DNA will be methylated. And as we separate the DNA from each other, the methylation stays with what it was originally on, which in this case is the blue piece of DNA. So our methylation is a signal because of this difference that we can see in the structure of the DNA, or the chemical nature of the DNA, after replication ends. As we'll see as we go back to the slide, eventually this is going to go back to this. So eventually both strands will be methylated, which means there's a short period of time where mismatch repair is actually going to be able to work. And there's more to the story, which is what we'll deal with on Wednesday. We're getting close to the end